Good afternoon, everybody, and welcome to the launch of the Fall 2024 IQB seminar series. It's great to see so many people in the room and online. For our first speaker, Michael Manhart, will be introduced by our colleague, Sagar Kare. Michael, can I have the next slide, please? So the program for the fall is laid out on this slide with one exception. There is no gap on the 16th of October now. Darren York very kindly pulled in a favor, and so we'll have a speaker on the 16th, and this slide will be, and the materials online will be updated accordingly. Next slide, please. Congratulations once again to Erica McCarthy, who received the 2024 IQB Inclusive Leadership Award in recognition of her outstanding achievements in training and mentorship. For those who don't know her, she's in the Darren York lab. Erica will be getting an all expenses paid trip to a scientific conference as part of the award to continue the kind of work that she obviously enjoys and is very good at. So next slide, please. Anyone who feels that either they should be considered for the 2025 award or would like to nominate someone for the 2025 award can do so by submitting an application on or before October 31st. The details are here, and if you need more information, you can talk to Michelle Sangera. Next slide, please. We have considerably beefed up the training activities that we're providing through the RCSB Protein Data Bank in response to the guidance that we received from our funders who called on us to de-emphasize education and emphasize training. So one element of that, a new campaign in training, are our virtual office hours. And the next one coming will be on October the 8th, 4 to 5 p.m. Eastern, 1 to 2 Pacific. It will focus on the use of advanced search on rcsb.org. I acknowledge freely that the advanced search page on rcsb.org is a little on the impenetrable. Once you figure out how to use it, it's incredibly powerful. We are taking steps to improve the usability, but nonetheless, if you want to actually understand how to get the most out of advanced search, Rachel is the person to listen to. She's our scientific support and customer service lead. Next slide, please. There remain opportunities for individuals working with the RCSB Protein Data Bank. My job is up for grabs, and a new advertisement has appeared in Science Magazine to that effect. And I hope that you'll encourage qualified individuals to apply. I want a fourth career, and I can't have that until I make an orderly transition of the leadership of the PDB to somebody who's going to be in the job for the next 10, 15 years and take it to even higher heights. We're advertising and interviewing right now for an IT infrastructure lead here at Rutgers. There are postdoctoral opportunities at UCSD, gap year and undergraduate summer research opportunities here at Rutgers. Depicted on the right are two of our 2024 RISE student software developers. The shorter of the two individuals, in fact, the shortest person in that picture, is now working with us as a gap year student. Ivana Truong, who's online, I see. Next slide, please. And I want you to be mindful of all the contributions to IQB operations that go on behind the scenes, and acknowledge the members of the team when you see them. So Sagar, over to you. Thank you very much.
Thank you, Stephen. Welcome, everyone, to the first seminar of the seminar series at IQB. It is my great pleasure to introduce Michael Manhart. Michael got a bachelor's degree at Stanford in mathematics and physics, and he worked on high energy physics. And then he made two excellent decisions. One was to move to biophysics, and the second was to move to Rutgers to work with our own Alex Morozov in physics here, where he did some really fantastic work on molecular evolution. He then moved on to Eugene Shachnowicz's group at Harvard University. Eugene is an expert in protein biophysics, protein folding, molecular evolution, and many other things. And then Michael moved from Boston to Zurich, where he was a junior group leader at the ETH. And then he joined the faculty in the Department of Biochemistry and Molecular Biology at the medical school in 2023. So it is my pleasure to welcome Michael and look forward to hearing your talk.
Thanks very much, Sagar and Stephen, for the invitation. Well, it's really nice to be here. I will try not to be too loud into the microphone. And so as Sagar said, I am an assistant professor now in the Department of Biochemistry and Molecular Biology at the Robert Wood Johnson Medical School, although my home is right next door here in the Center for Advanced Biotechnology and Medicine. So it's nice to come and see the neighbors. And today I want to tell you about one of the major areas of work in our lab, which is understanding how metabolic interactions between bacteria affects mutant fitness. And so I want to start with kind of a very broad motivation for this, since I think this is going to be different from what a lot of you probably think about on a day-to-day basis. You know, I think when a lot of us think about evolution, we think about this very long, slow process that takes place over millions of years. And for some things, that's true. But the thing that I think is really remarkable about microbes, as someone who is fundamentally interested in evolution, is that they can evolve incredibly rapidly in just days or weeks sometimes. They have this huge, enormous evolutionary potential. And I think one of the nicest demonstrations of this was this experiment published 8 years ago that many of you might have seen from the paper or online different places. And the way this experiment works is that they covered the top of this table with agar media that the bacteria can grow on. And there's these sort of zones on it with different concentrations of an antibiotic. And the way it works is that at the sort of end zones of the table, there's no antibiotic. And then there's these progressively higher concentrations moving towards the middle of the plate. And these numbers along the bottom here represent factors of the minimum inhibitory concentration for this drug. So in the middle, there's 3,000 times the concentration of the antibiotic that would normally kill these bacteria. And in the experiment, they inoculate the bacteria at both ends of the table where there's no drug first, and they see what happens. And so let me play this video for you. So they take a video of this from above. So you see the bacteria very rapidly grow on both ends of the plate where there's no drug. But as you can see, after just a very short amount of time, they start sort of sprouting these invasions into progressively higher and higher concentrations of the antibiotic, until by the end you see that they're growing all the way into the middle where there's this 3,000 times concentration of the MIC. And I think what's important here is to know the time scale of this video. This is obviously sped up, it's not real time, but this whole thing took place in a little under 12 days. So in just those 12 days, these bacteria were able to evolve resistance to this antibiotic to 3,000 times the concentration of it in that amount of time. And so that's really a remarkable amount of evolution that has to happen in such a short amount of time. So that's a laboratory experiment, but there have been other observations of extremely rapid evolution in natural environments as well. So I think one of the really nice demonstrations of this is from this paper that looks at Bacteroides fragilis in the human gut microbiome. And I'm sorry, I'm a little trying to figure out where best to point because I don't think the people online can see me. I think if I point over here, you can see me on the Zoom video. So what you can see here are different lineages of B. fragilis over time in the human gut. And at the beginning, there's basically like, well, if you look back even before they start sampling, there were these two lineages. At some point, one of the lineages starts sprouting these mutations that lead to new lineages. And then eventually one of them arises, this kind of light blue one here, which eventually takes over and excludes all the other lineages occupying its niche, until by the end of this, the tracking here, it's completely eliminated those other ones. And again, the time scale is what's important here, that this axis on the bottom here is days. So this whole thing took place in about 1.5 years. And so that's how much evolution happened in this person's microbiome over just about 1.5 years. So that's, of course, longer than 12 days, but that's still a very relevant timescale for a lot of things that we as humans care about. So the fundamental reason why this evolution happens so quickly in microbes, really boils down to the fact that they're accumulating just so many new mutations all the time. And this same paper did this really nice back of the envelope calculation to kind of give you a sense of what those numbers are like. And so what you can do is you can take the estimated number of bacterial cells in a microbiome, the mutation rate, the genome size, and then the number of cell divisions per day, and you can estimate that there's going to be about 1 billion to some trillion new mutations per microbiome per day. And so when you think, the microbial genomes are usually on the order of megabases or something, this is really a huge amount of new mutations that are just constantly, being sampled in these populations. So the question that this raises then is, what do all these mutations actually do? How do they change the cells in which they arise? And of course, you can ask that question at many scales of biology. These mutations will change things about proteins, gene expression, metabolism, as well as other traits. But if you're really asking about what's the evolutionary fate of these mutations, the thing that you really want to know is, how do they affect fitness? And for today, when I talk about fitness, I'm going to talk about it in the sense of the rate of change for a mutant's relative abundance over time. So if you were to measure the relative abundance of a mutant over some time, you would get some trajectory like this. And the fitness is basically the slope of this trajectory. So a mutant that would increase like this blue line here, we would call that a beneficial mutant. It has positive fitness. A mutant that doesn't change, we would say that's neutral, 0 fitness. And then a mutant that would decrease, like the red one, we'd say that's deleterious, it's negative. There's other issues about how you actually quantify fitness here that we just wrote a whole paper on, so I'm happy to discuss that later. But for what I'm talking about today, this is what you have to have in mind when we talk about fitness. So that's the fitness for a single mutant. What you really need to know, though, is the distribution of fitness effects across all mutants that could arise in the genome. And so if you had this information, you would put this in the form of a distribution like this. So you would have some kind of histogram, and you would see the beneficial mutants are over here, they're positive. You have neutral mutants in the middle, and then deleterious and even lethal mutations on the left side of this distribution. And this object, which is often referred to as the DFE, is a really fundamental object in evolution, because this is what you need to know to predict a lot of aspects of evolution that we would care about. So for example, you need to know the DFE to tell things like, how quickly will the population adapt? Will it adapt at all, or will it just accumulate deleterious mutations if there's not enough beneficial mutations over here? Will that adaptation occur in just a few genes or biological pathways, or will it occur across a wide number of different genes and pathways in the cell? And then also it can tell you how important different genes or pathways are in that particular environment. So for example, if you saw extremely deleterious or lethal mutations in particular genes, that would suggest those genes are very important in whatever that environment is. If you looked at a different environment and maybe they're not deleterious or lethal anymore, that would tell you those genes are not so important in that environment. So it also, besides these sort of evolutionary predictions that you'd like to make, the DFE also gives us insights into cell physiology and genomics. So the DFEs like that have been measured for many different organisms, not just microbes, but many different microbial species and in various environments. But this is one of the main challenges of this, that that DFE that I showed you would only hold in a particular environment. And then as soon as you change the environment, all bets are off, potentially. It could completely change. And just as a practical matter, we're never going to be able to measure these distributions of fitness effects across all possible environments. Microbes just face way too many different kinds of environments. And you can imagine any little change in that environment might change this distribution in significant ways. And so what that means is that we really need rules about how this DFE changes across these environments. So we don't just have to measure them every single time, but we can actually make predictions about how that distribution is going to vary across different environments, because that's what we would need in practice to actually make predictions for the evolution of a population. So one, of course, major component of the environments that microbes actually face in nature is other microbes. So if you look at any kind of image of microbes taken from a natural environment, you see something like this. You don't just see one type of cell all clumped together somewhere. You see this, very complex mix of lots of different kinds of cells, different bacteria, fungi, archaea, all mixed together in these environments. And so this is a major component of the kind of environments that microbes actually face in nature. This particular image, by the way, is taken from feces. But you see things in other parts of the human body as well as natural non-human environments. And so one thing that happens in these environments where you have different species or strains of microbes all together is that they can start exchanging nutrients. Some of them are secreting nutrients out, other ones are uptaking them and using them. And that process of exchanging nutrients is called cross-feeding. And in recent years, there have been a number of studies that have shown just how pervasive these cross-feeding interactions are in natural environments. So this is sort of a review survey done by a former colleague of mine where they tried to estimate how many cross-feeding interactions are occurring between different microbes in different environments. So on the left here, you're seeing a map of a bunch of different microbial species, and these are predicted interactions of cross-feeding between them. So basically, it's sort of everything talking to everything at some level. And then when you also look at the kinds of nutrients that they're cross-feeding, it's also kind of a little bit of everything. There's carbon sources, nitrogen sources, amino acids, vitamins, nucleotides, and potentially other things as well. So there's lots and lots of this exchange happening in natural environments. The other thing that, especially in the last year, I've become really excited about is the discovery that changes in these cross-feeding interactions have been found to be at least associated with a lot of human diseases, at least occurring in the gut microbiome. And so here is one of these studies that looked at this. So what you're seeing here is a table of different metabolites along the rows, and then different diseases along the columns. And each of these bars is a score that's basically the difference in how much metabolic cross-feeding is happening between a healthy person for that metabolite and someone with one of these diseases for that metabolite. And so a bigger bar means that there's a bigger loss of cross-feeding interactions under that disease. So for instance, if you look at IBD here, there's a really big change in a lot of these interactions for lots of different metabolites. So it might be, I'm sure it's hard to read a lot of these, especially out in the audience here, but there's carbon sources, amino acids, vitamins, lots of different kinds of metabolites that were detected here. And you can see there's other diseases like cirrhosis, that one's more surprising to me actually, that there's this big change in all these metabolites. But it varies across diseases. If you look at like arthritis, for instance, there's not as big of a difference. But it's been detected, and there's other papers from last year as well that are seeing these big differences. And whether this is, a cause of any of these diseases, that of course is a different question. But it's certainly something that's happening alongside these diseases in the gut microbiome. So changes in these cross-feeding interactions is something that is definitely happening in nature. Microbes are exposed to not just a constant environment in this way, but sometimes they see cross-feeding, sometimes they don't. So that raises the question that these cross-feeding interactions are changing in the environment of microbes. How do they affect mutant fitness? And then ultimately, how do they affect the evolution of microbes in those environments? I think it's helpful to think about two possible ways that these interactions could be affecting mutant fitness through the distribution of fitness effects. So one thing that these interactions could do is that they could change the overall shape of the distribution. And what I mean by that is they could shift it, so they could change like the mean fitness across mutations. They could also change the variance in it. They could squeeze it. They could change other kinds of shape parameters too, but you might think mean and variance are kind of the two most basic ones that you would think of. The other thing that they could do is change the fitness of individual mutations. And so, for instance, if you looked at a scatter plot of the fitness of mutants between an environment without an interaction and an environment with an interaction, this is about points that are sort of outliers that aren't falling along sort of a perfect correlation. So, for instance, a mutant that's over here has very low fitness without the interaction, but then has very high fitness with the interaction. So that interaction really changed the selection on that mutant. Or points down here would be the opposite of that. So these are two kind of levels at which interactions can change mutant fitness, and they're important for different kinds of questions that you would have about evolution. So changes to the overall shape of the DFE, this is important if you want to do things like predicting population level evolution, like just how fast is adaptation proceeding? You know, how many deleterious mutations overall will a population accumulate? Whereas the changes in the individual fitness of mutants, this is what you care about if you're trying to predict genetic level evolution. You want to know what genes are actually under selection, what mutations are likely to be fixed in that population. So this is this general question that's motivating this whole direction of our lab's work now, which is how these ecological interactions, particularly cross-feeding right now, affect the evolution of microbial communities. And so the kind of questions that we hope we're going to be able to answer eventually through the work we're doing on this are things like, do these ecological interactions cause a population to evolve faster or slower? Do they completely change what mutations even fix in one of those populations? And then in sort of the longer term of this kind of work, I think this has consequences for situations like in the gut microbiome or other human microbial systems, where, for instance, we know that stressed microbiomes often have lower diversity and fewer interactions, as I just showed you a few slides ago. So would that make those communities able to adapt more quickly to an environmental change, like, say, an antibiotic treatment? And especially if that turns out to be true, that's probably a bad thing, right? That not only is the microbiome already stressed because of the disease that's occurring, but now maybe it's going to be more able to adapt really quickly to something else, like, say, an antibiotic. So this is why I think this is the kind of question that's really important for a lot of other downstream things in microbiology and microbiome science. Okay, so what's known about this so far? So there have been a number of studies in the last few years that have tried to directly measure this. So they take a system, they look at a bunch of mutations in it, and they measure how does the fitness of those mutations vary with and without some kind of cross-feeding interaction. And one set of studies here has basically found that these cross-feeding interactions reduce the magnitude of fitness. So basically, they make everything just weaker. And the consequence of that, the reason why that would be important is because that would cause adaptation or evolution to be slower. And so, for instance, one of the systems that this has been studied in that was published last year, this is looking at cross-feeding between yeast and algae. This is not a natural community, but this is a synthetic system to study in the lab. And this is the scatter plot of the mutant fitness. Horizontal axis is, these are all mutations in the yeast, by the way, not in the algae. And so the horizontal axis tells you the fitness of these yeast mutants without the algae, and then on the vertical axis, this is the fitness with the algae. And as you can see, all of these mutants, or not all of them, but the vast majority of them are below the diagonal. And what that means is that their fitness is lower in the presence of the algae than in the absence of it. And so that's why they come to this conclusion, that this magnitude of the fitness effects is reduced. This was also seen in a completely different system in another paper published last year. So this is looking at cross-feeding between E. coli, as well as two other species, this Amextorkins and Salmonella. And again, these are all mutations in the E. coli. And you can see here, these are pretty much all deleterious mutations, unlike those. And so now we're looking at points that are above the diagonal, because these are deleterious mutations that are weaker in effect in the presence of the partner species than in the absence. And that happened in both combinations, with both other species that they were combining here. So some studies have found that cross-feeding tends to weaken fitness effects. Others have found other things. So this is another study that was looking at marine microbes in a model phycosphere. So a phycosphere is like the little ecosystem that forms around plankton in the ocean. And this was also a combination of 3 species. And the data is presented in the same kind of way. You have the fitness for the mutants by themselves along these horizontal axes, and then in combination with another species along the vertical axis. And basically, you don't see any clear pattern here. Some of the mutants have, stronger fitness effects. Some of them have weaker. And that's just kind of true in all the different combinations. So it's not really clear what the pattern is here. And this has been observed in some other systems as well. So kind of the situation that I would say this topic finds itself in now is that we definitely see that these interactions can change mutant fitness, but it's not clear how. What does that actually depend on? Does it depend on the kind of interaction? Does it depend on the species? Does it depend on other aspects of the environment? So now we're in this position where we know there's an effect, but we really need to figure out, is there a pattern, and what are the mechanisms behind that pattern? So this brings us to the work of our lab. And this is work that I started some years ago in my previous position in Zurich. And it was led in particular by a research assistant, Tomas, who was with me back then, and also Anastasia. And now here at Rutgers, it's being carried on by Athar, who's an undergraduate, and Duhita, who's a postdoc in our lab. So I'll be showing work from all of them today. And what we've tried to do is develop a model system for studying the effects of these interactions on mutant fitness that we think is more precisely defined than some of these previous systems, and will allow us to answer some of these questions more precisely. And so let me tell you what I mean by this model system. So in a lot of these previous studies, or all of them in fact, it's always about two different species, like the yeast and the algae, or E. coli and salmonella. And of course, in nature, that's probably what a lot of these interactions are. They're between different species. But the challenge with interpreting those results then is that whenever you put two species of microbes together, they do all sorts of complicated things. Like if you read all these papers, the story is like, we thought they were interacting in this way, and we started, measuring stuff and poking at them, and it turns out they're interacting in a bunch of different ways that we didn't anticipate. And that's probably real, what's really happening in natural environments, but it's also very complicated to interpret. And it doesn't, you don't really have good controls for that when your system is defined that way. So we've tried to take this completely opposite approach to really complement the data that's coming out of those systems. And what we've done is let's take a single species, E. coli, and let's actually engineer two versions of it to interact in one very specific way. So that way we know that that's the only thing they're doing. And a good candidate for this is taking E. coli strains and knocking out genes for amino acid biosynthesis. And so, for instance, we can take a strain and knock out this ILVA gene, and now it becomes an auxotroph for isoleucine, which means it can't make isoleucine itself. It has to get it from the environment. We can also do this for a methionine biosynthesis gene and make it an auxotroph for methionine. And then what happens is if you grow them by themselves, you have to give them that amino acid that they can't make. But if you grow them together, now this isoleucine auxotroph will secrete some amount of methionine. The methionine auxotroph will secrete some amount of isoleucine, and they can actually complement each other in this way, and they can grow together through this cross-feeding interaction. And the reason why we focus particularly on these amino acids here is that amino acid cross-feeding has been found to be very abundant in natural environments. And in particular for these two amino acids. So here is a plot that just shows you the prevalence of auxotrophies for different amino acids in natural environments. And basically you can see like this set of amino acids are very common, including isoleucine and methionine, but other things too, like tryptophan is very common, leucine, and some other ones. And so we think this particular interaction is something that is occurring a lot in nature. And so the biology of it is a good place to start to learn about this problem. So that gives us our cross-feeding interaction. Now what we need are mutations. And we do this using this technique called transposon insertion mutagenesis, which has really become very well developed in the last several years. And the way this works is that you take a background genotype, so say our isoleucine auxotroph, and now what you do is you can make this library of transposons. And what makes it a transposon here is really just that it has these particular inverted repeats at both ends that are recognized by a transposase. But the trick to this is that you put in these barcodes inside that transposon that are going to allow you to sequence it and identify it very easily. And now you make this library of all of these different transposon DNA fragments, and you can then get those into the cells, give them the transposase, and that transposase will now insert all of these transposons randomly into the genome. So this works with a transposon called TN5, and it has very low sequence specificity, so it just kind of goes randomly in the genome. And so every cell that goes through this process will get one of these transposons inserted randomly in its genome. And what that means then is that the insertion of that transposon we use as like a mutation. And in particular, because these are bacteria, their genomes are mostly coding regions. So mostly these transposons end up inserting in genes, and mostly those end up just knocking out the gene function. And so we think of this as a proxy for like loss of function mutations that would occur spontaneously in genomes. So they're not exactly the same as other kinds of spontaneous mutations, of course, but we can at least use them as a model for these loss of function mutations. And at the end of the day, the good thing about this is that you can do it very efficiently. You can get tens or hundreds of thousands of unique mutants this way pretty easily. And here on the right, this shows the distribution of these insertions across the genome. As you can see, it's pretty uniform. And so this is number of insertions per 10 kb. And so you can see you're getting 10 to 1,000-ish insertions per 10 kb. And so that means most genes have a bunch of these insertions within them across all the cells in your library. So altogether, this gives you this library that you can make on the background of each of these genotypes with tens or hundreds of thousands of unique mutations. And we've done this for all of the strains. So our two oxytrophs for isoleucine and methionine, and then also we have the ancestors of them that we use as controls. So this is how the experiment works. Now it's pretty simple. We take this library of all these mutations, we grow them by themselves for one of these oxytrophs, and we do this by serially passaging them every 2 days. And then we now want to know, how did those mutations change in their relative abundance? Because that's what we need to measure their fitness. So we sequence these barcodes that label every transposon at every time point, and we get these trajectories like I showed you at the beginning. And then, like I said then, the fitness is basically the rate of change of these trajectories for the mutants. And then that allows us to get this distribution of fitness effects for these mutations without the cross-feeding interaction. And then we just do the same thing, but in a culture with another strain. And so now we take libraries for 2 different strains, the isoleucine oxytroph, the methionine oxytroph, grow them together, we do everything the same, and now we get the distribution of fitness effects in the presence of the cross-feeding interaction. And then finally, we can combine these 2 things to compare them and see how did the DFE and the individual mutant fitness change with and without this interaction. So that's the experiment. Let me show you the data that we've gotten with this. So the first thing that we've seen, so if you remember, I told you that the interactions can change mutant fitness in 2 different ways. It can change the shape of the distribution, it can shift it, it can squeeze it. It can also change the fitness of individual mutations. So the first thing we've seen is that the cross-feeding changes the shape. And basically, we see that it starts to shift this distribution. So what you're seeing is in red, the distribution of mutant fitness for in the background of the isoleucine oxytroph. So this is this distribution of fitness effects. And the red is without the cross-feeding interaction, and the blue is with the cross-feeding interaction. And what this shows is that basically the cross-feeding has shifted the DFE towards higher fitness. So there's fewer of these really deleterious mutations over here, and more of these quite beneficial mutations over here. And we see something extremely similar, actually even a little stronger, for the methionine oxytroph. When the methionine oxytroph is cross-feeding methionine, it also just shifts this whole distribution to the right. So that would suggest that adaptation is going to be a lot faster in the presence of these interactions. The distribution here is kind of centered around 0, more or less. So there's kind of an equal number of beneficial and deleterious mutations. So there might not be a lot of adaptation. But in the cross-feeding case for both of these strains, now they really got some significantly beneficial mutations that weren't there before. So that means there's going to be much stronger adaptation happening because of that cross-feeding for isoleucine or methionine. But we also see changes in the fitness of individual mutants. And I think the easiest way to look at that is to look at trajectories of some individual mutants. So if we look at the isoleucine auxotroph and we look at all of its mutants in the presence of cross-feeding, we see that there's these two mutants that are really beneficial, and they really increase in frequency quite rapidly. So one is an insertion in the PTSG gene, another is in the gene RPOS. And what's interesting is now you can look at those same exact mutations in the absence of cross-feeding, and you can see they're either deleterious or maybe the RPOS one is kind of neutral. So this cross-feeding interaction has completely flipped the sign of the fitness effect for these particular mutations. And for those mutations, we see the same thing in another replicate. They were also very beneficial with cross-feeding and neutral or deleterious without cross-feeding. The reason I'm showing the second replicate here is that the weird thing that happened was there's this other lineage that has insertions in this, what I think is, as far as we know, believed to be like a prophage, YFJI, that doesn't really do anything in this replicate, but completely takes over in the second replicate. And as you can see, it doesn't do anything in the absence of cross-feeding. So we think in that case, there's actually some secondary mutation happening, but we're in the process of sequencing it and figuring out what's going on with that. And we see something very similar for the methionine auxotroph. So again, say there's these two major beneficial mutations, one in MADC, one in METJ, that are consistently beneficial under cross-feeding in both replicates, but then without cross-feeding, or deleterious or neutral. So again, that interaction has completely changed the sign of these mutations. Now, what was interesting here is I told you about this one replicate for the isoleucine auxotroph that has this prophage insertion that does something bizarre. And so actually in that same replicate, there's a mutant in this gene PERU that also does something bizarre that's not replicated. And so if you put those side by side, what you can see is that, these are the two this is replicate 1, but for the methionine auxotrophs, sorry, that's a typo. That should be methionine auxotrophs, methionine mutants, and then these are the this is isoleucine and this is methionine. And you can see that sort of these two mutants are kind of matching here, but then this, other strange one, this YFJI here and this PERU one are co-occurring in this second replicate. And that raises the possibility of whether, whatever this mutation here is, it's not just sort of generally good under isoleucine cross-feeding. It has something to do with another mutant and the other strain also, rising to high frequency. So somehow their fitness is dependent on each other. And that's, again, something we don't know anything about what's really going on there, but the data suggests that it's not even just a mutation is beneficial with cross-feeding, but it might also depend on the specific sort of genetic context of other mutations that are present there at the same time. So that gives you just an overview of the data that we've gotten from this experiment so far. And now I want to tell you something about what's actually causing these effects. What's causing cross-feeding to change the fitness of mutations. And this is where I think modeling is really valuable, computational modeling. Because when we work with models, we're able to really control everything that we can't do in an experiment, but also we can basically, tear it apart and understand every little thing that's going on because, it's something that we can do on the computer. And so this has been work led much more recently than the experiments by a former graduate student of mine, Eustace, and an undergraduate here at Rutgers, Drew. And for this, we've been using this modeling approach called whole genome metabolic models. And I want to tell you kind of briefly about how these work. So the idea of these models is to reconstruct the entire metabolism of a cell. So what you do is you start with a list of every metabolic reaction in a cell that you know about, and you put this in the form of like a big stoichiometry matrix. And now the sort of important part of this is now there are fluxes through all these reactions, which could go forward or backward. And the idea is that we're looking for something in steady state, so where all these reactions are in some steady state. So that's why you would have this matrix times the fluxes ends up equaling 0. And now you force this mass balance condition. Sometimes there's other constraints too for sort of other considerations. But then ultimately what you want to do is you define some kind of objective function, which is really the growth rate of the biomass. And this is usually defined by saying that the composition of the cell has some particular stoichiometry of like every amino acid and every nucleotide and some other components. And so those the rate at which those components are being produced is the growth rate of the whole cell. And since the way you define the system, it's not completely, this is not a well-defined, mathematical problem. But what you can do then is assume that whatever gene regulation is happening inside the cell, it's doing in a way to maximize this growth rate. So then you can subject these constraints to this optimization condition, and you can figure out, A, what is the growth rate given whatever this metabolic network was, and B, what were the fluxes through all these reactions for that growth rate. And so that's the outcome of this optimization algorithm that you run. And so what we can now, this is basically a little black box that allows us to now test how would mutations in the metabolic network lead to changes in growth rate. And then that's what we use to predict changes in the fitness. And the way we do this is now we kind of just reproduce this experiment that we did in the lab, but in the context of this model. We take a focal strain like this isoleucine oxytroph, we knock out every metabolic gene in its genome, and then we calculate the growth rates of all those gene knockouts in a monoculture where there's no cross-feeding. And then we do the same thing, but in the presence of the cross-feeding. And kind of the important detail here is that these mutants are growing in the presence of whatever the partner strain is, secreting out into its environment, but also whatever their ancestor is secreting out into their environment. Because these mutants are very rare, right? They're arising just single cells at a time. So when they look out into the world, they see basically everything as the partner strain and their ancestor. And so they see whatever both of those two strains are secreting out into their environment. And then that creates the environment for these mutants. So we can calculate the fitness across all these mutants, and we can do the same kind of analyses that we did with the data. And here's what we see. So along the horizontal axis, this is the fitness of these gene knockouts without the cross-feeding. And then on the vertical axis, you see the fitness with cross-feeding. And the salient feature here is that you see a bunch of mutants sitting right up here along this line. And these are mutants that, without the cross-feeding interaction, were quite deleterious. They have all these negative fitness effects. But now, in the presence of the cross-feeding, they're all sitting at 0, which means they become neutral. So in the parlance of evolutionary biology, we would say the cross-feeding interaction has rescued these deleterious mutations. It's brought them back, and now they're no longer so bad anymore. So it turns out, we're knocking out every gene, so there's, 2,000-ish genes here that we can knock out. We find 44 of those are these deleterious mutations that get rescued by the cross-feeding interaction. So the question now, like I said, the value of the models is we can really figure out what's going on. And so now we can first ask, well, what's actually changing in the environment with and without cross-feeding these mutants are seeing? And there's basically two things changing. So here on this axis, this is like the uptake flux in the environment. It's like the availability of a nutrient in that environment that the cell sees. And so we can first look at the availability of methionine in that environment. We're going to focus on the methionine oxytroph right now. And you can see that, so the light blue is without cross-feeding. So there's a lot of methionine there when they're not cross-feeding. But now when they're in the environment of cross-feeding, the amount of methionine available goes way down. And that's because it's not being just given to them on a silver platter. They have to sort of slowly take it up from the other cell that's, producing it while also trying to grow itself. It needs methionine too. So one thing is this quantitative change in how much methionine is there. The other thing that changes is there's a bunch of other nutrients that are there too that weren't there before. And in particular, you see, well, there's all this isoleucine because, of course, the methionine oxytroph has to be secreting isoleucine for the isoleucine oxytroph to consume. And so the mutants are also seeing that. They're seeing a bunch of isoleucine there that wouldn't normally be there if they were just growing by themselves, as well as some smaller amounts of some other things like glycine and histidine and adenine. So the first thing that we see is changing here. I want to focus on that first one, so the change in the methionine concentration. So what we can do is we can look at all of these different mutants, and we can look at how their growth rates vary as a function of the available methionine. And what we see is that they have this dependence. If you remember, like Michaelis-Menten, this is sort of like a Michaelis-Menten dependence, except it's purely piecewise linear. But you can see when there's low amounts of methionine, growth rate goes up linearly, and then at some point you get enough methionine, and then the growth rate doesn't increase anymore. And what we see is that this pink line is the ancestor, and all these gray lines are all these 44 rescued mutants that I showed you earlier. And you can see that what happens is that when we go to cross-feeding, that reduces the amount of methionine in the environment sort of across this critical threshold, such that when you had a lot of methionine, you were up here, and all of these mutants were not growing as well as the ancestor, and that's why they were so deleterious. But by lowering the methionine, you've really shifted them into this regime of being methionine limited. And now whatever the differences in those mutants are doesn't really matter anymore. I mean, the way I thought about it is like, if you have a paper cut that hurts, but if someone, like stabs you with a sword, the paper cut's not such a big deal anymore. So now you're really low on methionine. You don't care about whatever other things, were bothering you before. All you care about is the methionine. And in that case, now all the cells, all these mutants and the wild type are equal to each other. And so it turns out that explains a lot of the rescue. So we can basically then check the fitness of all these mutations just in an environment where we've lowered the methionine, but we didn't change anything else. And we see that that explains 36 of the rescued mutations out of the 44. So that's most of it, but there's this other 8. So what is rescuing those other 8? So now we can take all of those other factors in the environment that were different with and without cross-feeding and add them in one by one and in combinations. And what we see is that if you add in the adenine, the glycine, the histidine, they don't really do anything. They don't really rescue anything by themselves. And also the isoleucine by itself doesn't rescue anything. But you get this 44 total if you add all of these things. But now it turns out that if you have the combination of the lower methionine and the isoleucine, that's what gives you all of the rescue back. Whereas the lower methionine and these other things, that doesn't do it. And so what's happening here is that these mutants, these other 8 rescued mutations, are apparently cheaters. That they're not only cross-feeding the methionine that they always needed there, but they're also just consuming the isoleucine that their ancestors produced. And so that's what allows them to be rescued. If they were just by themselves, that wouldn't work, right? There's no isoleucine around to consume. But in the presence of the cross-feeding, now they can take advantage of that as well. That creates this other niche that they can occupy. And so that's the second mechanism besides the change in nutrient limitation, but also this new niche that is available for cheating off your ancestor. So with that, I want to summarize what I've told you so far today about this work. So the really broad point that I hope you remember here is that mutations in microbes are extremely frequent. There's tons of them all the time, and they can lead to really rapid adaptation, both in laboratory environments as well as nature. And these ecological interactions between microbes are also very frequent in nature, and they can change under diseases, under changes in the natural environment. There's ideas about how climate change is changing these interactions in nature. And we know that those definitely have an effect on mutant fitness, but we don't know what the pattern is. And so what we've done is we've tried to focus on this model system where we can really precisely control interactions to test exactly this kind of interaction leads to this change in mutant fitness. And so what we found is that cross-feeding isoleucine and methionine actually have very consistent patterns. They tend to shift the DFE towards more beneficial mutations, and they also change the selection on individual mutations, which means that they would both change just how much adaptation occurs in those populations, but also which mutations are actually going to be driving that adaptation. And then finally, by using these metabolic models, we predicted that there's these 2 potential mechanisms by which the interactions can change mutant fitness. They can change the limitation state by making them more limiting for that nutrient that's being cross-fed, and then that changes selection on these mutations. They can also create a new niche for these cheaters to arise by taking up nutrients from their ancestor. There's certainly other possible mechanisms as well that we've been thinking about, but they're not realized in this model, but that's something that we certainly will look for in the data as we dig into it more. So I want to stop there. Thank you so much for your attention, and I'm happy to discuss further any questions you have. Yeah, Sagar. Oh, do you take this one? We could just.
Yeah.
No problem.
Yeah. Thank you, Michael. That was really cool. My question is sort of, I have 2 questions actually. So one is about if you're co-culturing organisms that grow at have very different doubling times, how does that affect some of these things, right? Because things might be produced at different rates, and things are. So adaptation might occur on different time scales. So have you thought about that? And then the second one is, if microbial adaptation is so rapid, right, like what you illustrated in the movie at the beginning, then why does antimicrobial resistance take so long to appear, right? It takes like decades and so on to really spread. So yeah.
Okay. Is this one working now? Yeah. So for your first question, Sagar, are you asking about two species or strains that are growing at different rates at the same time together? Or are you talking about, does the growth rate change between with and without the interaction? When they're together. Yeah. That's potentially very important. So in the system that we have, they have to grow basically at the same rate, because I mean they're so similar for one thing, but also, they got to exchange, at least when they're at the balanced sort of stoichiometry of the two strains, they grow together at the same rate. Yeah, I think that could make a huge difference. I mean, also just we know that a lot of things about cell physiology change a lot with growth rate. I don't think I have a simple answer for you, but I would agree that it changes a lot. But I don't know how exactly it would change, but I think it's important and something to look at for sure. Your second question, why is antibiotic resistance not even worse? Well, that's a very good question. I think there's more qualified people than me to answer that around here, even at Rutgers. I mean, to me, the simple answer that I would give from the evolutionary side is that usually resistance carries some cost. That you're carrying an extra gene, you're making something, you've knocked out an efflux pump that now is bad sometimes without the drug. And so I'm sure that plays a role, that you don't get it for free, that they can get the resistance, but then that's bad once the drug is gone and there's some other environment. But I don't really know if there's a more other mechanisms too. I mean, I think there's definitely bottlenecks as well, that you can't always get the mutations that easily for certain drugs. Sometimes you have to get horizontal transfer of a whole gene. So it might also vary for different drugs and for different species.
Okay, I have a question. So I'm just wondering why you chose isoleucine and methionine for your models and not another amino acid.
Right. I mean, I think, part of it was just these are sort of strains that were already well characterized, and their cross-feeding was already well characterized. But it's also known that those are, so some of it's that they're good at cross-feeding. Not every amino acid can be cross-fed very well, because they don't get produced enough, or they don't, go in and out of cells very well. So some of them just don't cross-feed very well. Methionine and isoleucine do. But also, I guess probably as a consequence of that those particular amino acids are known to be cross-fed a lot in natural environments. They're not the only ones. There's other ones as well. And so that's one of our goals in the future, is to try other amino acids, tryptophan, proline, for instance, leucine, lysine. And those are also common, and they may have differences in their effect on mutant fitness. But these were, we thought, a very good starting point to start working on this for that reason.
Yeah, I have a question about your very nice experiment. The experiment in your experiment, you used the two mutant species that are kind of dependent on each other. In kind of the interaction or coupling is kind of strong, right? Because without one, the other is going to be, you're not going to survive well. But what if you loosen the interaction? For example, use wild type for one species, and the other species is kind of a mutant, lacking isoleucine or methionine. Then, in that case, the fitness landscape, for example, is gonna change in different way than what you showed today.
That's a great question. Yeah, we have not done those experiments, but that's something we want to do. I mean, the way I would put that is we're studying obligate cross-feeding. They have to cross-feed. You could also study facultative cross-feeding, where they can, but they don't have to. And we have not done that. Another thing you can do, actually, that's also, I think, interesting, especially to test this idea about how important is just the change in methionine concentration, is you can still put the two oxytropes together, but you can give them a little bit of methionine and like titrate that over some range. And you can see, if you give them a whole lot, do they behave as if there was no partner there at all? If you give them some intermediate amount, does it sort of interpolate between the two things? Yeah, we haven't done those experiments, but those are really interesting things to really understand this better. It's a great idea.
Thank you for the talk. This is sort of what seemed to be a minor point because you didn't spend much time on this, but you were talking about disease conditions and how that affects cross-feeding of specific nutrients, how much is affected. Are there any, I'm not sure I caught this, are there any disease conditions in which you have less cross-feeding of nutrient A, and a lot less cross-feeding of nutrient A, but you have more of nutrient B because of that disease condition? So it's really just shifting from one nutrient to another.
As opposed to just everything going up or down together. That's a good question. I didn't think about that. We would have to look through the data in those papers more. I imagine you'd find at least some case where that happens, just because I think there's, everything happens somewhere kind of. But whether that is common or not, I don't know. That's an interesting case to look at. I would also say a lot of those predictions about individual cross-feeding interactions, I would say by themselves are not necessarily all that reliable because they're mostly done on the basis of metagenomes. So generally, I would trust sort of the overall effect of like the cross-feeding is broadly going up or down. I would say for any individual one, you'd have to go in and actually do targeted experiments with that. I wouldn't trust it just on the basis of the data that people have been publishing so far. But you could look for ideas to start.
Yeah. Thank you very much. That was a great talk. I did want to ask a question of my own. There aren't any, despite the fact there are as many people in the room as there are online, there were no questions open online. How long does this ancestor effect last?
What do you mean by ancestor effect?
Well, the cheating off the ancestors.
Oh, I don't. Yeah, I mean, it can't last forever.
No.
Yeah. So I mean,
eventually
yeah,
the older bugs are going to die, and then whatever advantage they're conferring will evaporate.
Yeah, that's something we need to test. So in the way we do the model is sort of like it's like a snapshot in time, just when the mutants are very rare. So we need to do a different kind of simulation to sort of extrapolate as they would increase in frequency. Where did the diminishing returns set in, and they start? Does the community collapse, or do they reach some steady state? Right. So, and also we need to test to see if that's actually happening. We think that could be happening in some of the mutants we see in the lab, and then we would need to run that experiment for longer to see that. So,
right.
So I don't know, but yeah, that's an important question.
So I thought it was, and maybe we could just go back to your slide where you showed that the changes as a function of different disease states. It was right. It was during your introduction.
Yeah.
As a clinician, I found that very interesting.
This is not the only paper on this too. There's been several in the last year and a half.
Yeah, so you've got inflammatory bowel disease, cirrhosis. What's ANK?
Ankylosing.
Spondylitis. Okay.
Yeah.
And then there's non-alcoholic fatty
fatty
acid liver disease.
Behçet's, chronic fatigue syndrome, type 2 diabetes.
Type 2 diabetes, yeah. I was surprised that type 2 diabetes.
I think that's
colorectal cancer, that's what
cancer,
CRC stands for. Now, I was surprised that type 2 diabetes has so few impacts. Anyway.
There's other papers. Yeah, I also, like I was in the answer to this other question, I feel like these are also statistical analyses. You should probably look at multiple ones before you decide anything too clearly. So
we need a meta-analysis too.
Exactly. There was a really great one that I didn't put in this one where they looked at, people are always claiming about how diversity of the microbiome changes across diseases.
Right.
And I found this really nice review where they did a meta-analysis across lots of studies and lots of diseases. And they show that some diseases do that, like IBD is the classic case of diversity going down.
It's obvious why that would be the case.
But there's lots of other ones where they don't. And they look at all sorts of weird things like HIV infections, and I think also diabetes is in there. And it's a very mixed outcome for a lot of diseases. It's not clear.
Okay. Well, we have some PDB swag for you. The audience knows what's in the bag. You will discover for yourself.
Thank you very much.
Thank you very much. And I ask you all to join me in thanking the speaker.
Thank you all.